In exercises 8 and 9 of your lab manual, we look at two special stains, which are stains that are done in very specific circumstances. We're going to look at the capsule stain in exercise 8 and the endospore stain in exercise 9. Because of the technical challenge of these two, as well as some risk, we don't actually perform these stains in microbiology, but we do want you to know about them and be able to look at some stained images that are already performed and understand what you're looking at. So that's the goal today, and there are some questions in some places in your lab manual where you can draw these examples out, so I encourage you to do that. So starting out with exercise 8, the capsule stain, here we see the method in which this is done. This uses an acidic dye. Remember, acidic dyes are generally repelled by the inside of the cell and stain the background of the image rather than the cell itself, although there's usually a counter stain done as well to stain the inside of the cell. So this would be a great example of what we would consider a negative stain, because as you can see when we look at these pictures, so this person is putting a drop of dye and a drop of bacteria at the beginning of their slide, and then very carefully, and this is the technically challenging part, using another glass slide to spread that very thin. And what we're left with, especially when we do a counter stain, is the background is stained. That's the definition of a negative stain. The background is stained. When we counter stain with another dye that can stain the inside of the cell, we see the white image around each cell. That white image is the capsule. So that's a mucoprotein layer that's very thick that doesn't really take up dye, and that's why it's left looking white around these cells. So that is the capsule. Remember, a capsule is a virulence factor, does a couple things for the cell. It protects it from phagocytosis, so it's harder for our immune system to destroy encapsulated bacteria. It also helps the bacteria adhere to its environment. So these are very sticky and very difficult to eliminate. Here are a couple more images. We see here on the left side a little higher magnification. This helps us see just how thick these capsules are, because what we're looking at here, the dark purple bacillus, that's the actual bacterial cell. You can see the capsule is actually quite a bit bigger and thicker than the cell itself. And then our capsular stain is the negative stain that's staining the background, so we can see nicely the outline of a capsule. Here's another set of two images. I specifically chose these because the people who made these stains use different colors, and I don't want you to focus on specific colors, because oftentimes we can use different color dyes for the same purpose. So we see here a light blue negative stain and a pink negative stain doing exactly the same thing. So I would encourage you to look at these pictures, as well as perhaps do a Google image search for capsule stains and fill in the examples in your lab manual, and then also make sure you know what the function of a capsule is. Exercise 9 talks about another special staining procedure called the endospore stain. So let's talk first about what an endospore is, and let's remember that very few species of bacteria have the ability to make endospores. So this is something that is very specific to two major groups, the Clostridia and the Bacillus genuses. The idea of endospore formation is whenever those particular groups are stressed by, let's say, dehydration or temperature or some other environmental insult, they can very quickly form protective structure around their DNA. So this is a review from lecture material about how that happens. This is the process called sporulation. And I want you to remember these endospores are very different than the spores in fungus. So spores in fungus are there for reproductive purposes. These endospores are not reproductive. They are specifically for self-preservation. So we see here, as soon as this cell is insulted by some environmental stress, it copies its DNA, which looks very much like it's trying to reproduce, but remember, it's not. And then as the DNA is replicated, the cell membrane starts to fold in, and you can see here actually two separate sides to this cell forming around each set of DNA. We call it a forespore. And then as we move toward a more mature spore formation, one of these is going to become the spore, one set of DNA. The other set of DNA is actually going to fall apart. And we're going to keep wrapping material around this live set of DNA to protect it. Okay, so here we see that set of DNA is now completely shredded up. There's some calcium and some proteins forming a cortex, or a thick coat, around one set of DNA. That coat's going to keep getting thicker. Actually, all this DNA is going to actually throw itself onto the coat. So we're just kind of piling everything but the kitchen sink around one set of DNA, making that coat thicker and thicker. And then ultimately we have a mature spore. Now these endospores can remain viable. They're hardly metabolically active, so it would be hard to call it a live cell, but they can remain viable for sometimes hundreds of millions of years. So recently there was a fossil found with 200 million year old bacillus endospores that were still viable. So once these spores are put into the right setting um, to open up, then they can become again a vegetative or viable living cell. A couple of things you should know, a couple of terms. The position where these endospores form can be important, so I would like you to know these three positions. In example 1 here, and example 4, and example 6, we see the endospore forming roughly in the middle of the cell. And you notice here they're all pretty much bacillus-shaped bacteria. We call those a central endospore. So the reason this is a potentially valuable thing to realize is because we might be able to learn what we're looking at by looking where these endospores form. Examples 3 and 5 are forming their endospores very much at one end of the cell. We call that a terminal endospore. Number 2 is kind of a weirdo. We call this a subterminal endospore former. In these guys, the spore can kind of form anywhere. That would not make a very fair test question. So you can imagine if I'm showing you pictures of spore formers on your lab exam, it's probably going to be the central or terminal examples just for clarity. But I do want you to be aware of these subterminal guys, and this little diamond here is supposed to represent the movement that the spore can have anywhere between pretty much central and pretty much terminal. The process of doing an endospore stain is technically challenging and also potentially dangerous. So we don't do it in the NWTC microbiology class. It requires us to steam these endospore forming bacteria. That's actually what triggers them to form endospores, and also what allows them to take up dye. So we see here a Bunsen burner. We probably would use a hot plate with a steamer, and our slide is set on steam. That has the potential to get these endospores into the air and for us to breathe them in. So that's why we choose to not actually perform this, but we do have you look at completed examples. For example, here are some images that I've pulled from the internet that I think make good examples of what we're looking at here. And notice the endospores themselves are stained a different color, so this is a differential stain, and we can see in this example the endospores look like they're very much swollen ends of the bacillus, so we would probably call those terminal. Over here, the endospores are stained green, which is pretty typical, green or dark blue. The endospores look like they're subterminal, they're not quite central. And here's another image where these guys look pretty much central. The endospores are the green structures, the rest of the cell is stained red. So I would encourage you to complete the questions and definitions in exercise 9. Draw some examples. Probably might be helpful to do a Google image search and find a couple more examples of these endospore stain structures. And then of course complete your questions.